and identifying all of the things that are secreted and reabsorbed all along the length of the nephron. Here we have our filtration, and here we have all of the things that are reabsorbed back into the proximal convoluted tubule. These were all filtered out. They were filtered out through those fenestrated capillaries in the renal corpuscle. And then you see that there is some still secretion happening in the proximal convoluted tubule. One of those, very interestingly, is urea. And you see that we have our nitrogenous wastes, some drug hydrogen ions. It goes down through the loop. And this thin descending portion of the loop, remember, it's permeable to water, but not much else. We can see there's a little secretion of urea into the tubule from the blood and from the surrounding tissues. Then we come up the ascending side and we see that we have reabsorption of sodium, potassium, and chloride. These two are a driving force for that water conservation, the big part of what the kidney is all about. And then there's this portion here about the urea. And I want to go to a little video to set us off. Quick question, it's really valuable, yeah?
So for this diagram, I guess the words reabsorption and secretion and the arrows are like messing with my brain.
Oh.
So the secretion is going into the tubes,
Correct.
Reabsorption is going out of them.
Out of the tubule,
Out of the tubules.
back into the interstitial fluid,
Okay, so
and ultimately the blood.
okay, so this interstitial fluids are reabsorbing what the tubules are secreting. So
No. So secretion is coming from the blood into the tubule.
okay,
It has to, remember, there's fluid around everything.
yeah,
There's nothing here, right? Except for squamous cells that are attached to each other and columnar cells that are attached to each other. Epithelial cells,
yeah.
no tissue, no fluid around those cells. But here we have fluid around, and then we have this peritubular capillary bed that's surrounding all of this. And unfortunately, many of the diagrams don't give you that peritubular capillary appearance.
Yeah.
But you have to remember that we
there
have
is
blood
one.
supply all over all of these tubules, because this efferent arteriole becomes peritubular capillary.
Yeah.
And so we're going to be sending water, is going to be reabsorbed from the tubule into the surrounding
Okay.
tissue and then the blood ultimately.
Okay, I guess my brain was thinking, it says tubular reabsorption, so I was thinking the tubules are the things that are reabsorbing, but it's
Yeah, so
No.
tubular reabsorption is from the tubule into the
to
blood.
okay,
And tubular secretion is from the blood into the tubule.
okay.
Yeah, I know.
I'm gonna have to reword that in my head somehow, but
Yeah. So, back to my one, that little video. We're going to watch that video together because this is where we're headed today, is to talk about. I should have queued it up, but I didn't.
Is the body's filtration system and produces urine in its thousands of nephrons, the kidney's basic unit. Each nephron begins with a boneless capsule wrapped around a capillary ball, the glomerulus, which becomes a tubule that eventually forms the loop of Henle, before collecting into tubules and ducts that empty into the bladder. Fluid from the blood enters the nephron by filtration and passes along it, where substances are added from the blood by secretion or removed from it by reabsorption. Potassium and hydrogen ions, organic acids and bases, metabolites and foreign substances are secreted across a selective membrane. The potassium and hydrogen are exchanged for sodium along the tubule. 99% of the filtrate is reabsorbed through a mixture of passive diffusion down an osmotic or concentration gradient and active transport by pumping against a concentration gradient. The kidneys can conserve water. The only way that the body can move water is by moving sodium chloride first. You have to create a concentration gradient so that water can move by osmosis in the direction of the highest concentration. The loop of Henle creates a hyperosmotic region in the medulla of the kidney, acting as a sponge, drawing water out from the collecting duct, if the duct becomes permeable under the influence of vasopressin, also known as antidiuretic hormone, or ADH. The ascending limb of the loop of Henle, which is impermeable to water, pumps out sodium chloride via sodium-potassium chloride co-transporters from the urine into the blood in the vasa recta. Blood in the vasa recta carries this sodium chloride into the medulla, making the entire region hyperosmotic. The blood, which is moving in the opposite direction to the urine in the loop of Henle, then ascends parallel to the descending limb of the loop of Henle. This descending limb is water permeable, allowing water to move out of it into the vasa recta. This entry of water into the ascending vasa recta reduces the osmotic potential of the blood to normal as it returns to the body. The interplay between the loop of Henle and the vasa recta is called the countercurrent multiplier. Essentially, the medulla acts as a sponge, retaining and releasing water to address the water requirements of the body. ADH acts on the collecting duct to make it permeable to water, allowing water to move into the hyperosmotic medulla, where it is drawn up by the vasa recta and returned to the body.
So that's one mechanism by which we can study how the kidney is functioning, and this is what we're actually heading toward and talk about today, is that countercurrent multiplication aspect, and how the kidney concentrates the fluid that's leaving the body via urine, as urine, right? And conserving as much water as possible. And we talked about how the kidneys have two different kinds of nephrons. There's the cortical nephrons, and there's the juxtamedullary nephrons. And the one that he was talking about was the juxtamedullary nephrons, because their primary job is concentrating urine, conserving water. The cortical nephrons are all about filtering out the toxins, the old hormones, the whatever, urea, everything, get rid of all that stuff that we don't want in our blood supply, is the cortical nephron's job. And so we're going to head toward this discussion about the countercurrent multiplier system of how the kidneys function. So, as you can see, along with these guys in the proximal convoluted tubule, lots of reabsorption here. Not as much reabsorption in this convoluted tubule. Some of the important things to note is that we're going to be secreting hydrogen ions in multiple places. We're going to be secreting urea in multiple places. We also see this urea and reabsorption of water here from the collecting duct. This urea is part of what he was talking about, the medullary sponge. So it serves as an osmotically active particle. That's why it's being moved around quite a bit. It's being put into the tubule, and then it's being pulled out of the tubule and back into the tubule, and its job is to draw water. And then ultimately get rid of it, because urea is one of those nitrogenous waste products that we don't want to carry around with us, right? So another important point to note is that while we are reabsorbing sodium, potassium, and chloride here, notice that we are now kicking that potassium out of the circulation here. And the reason for this is because we have to maintain that intracellular level of potassium and maintain our extracellular level of potassium at very low levels. And once we start talking about fluids and electrolytes, you're going to appreciate how incredibly dangerous high levels of potassium are that are not contained within a cell. This is another really excellent page that describes what happens in the different regions, the proximal convoluted tubule, the renal corpuscle, the distal versus the loop versus the principal cells and the collecting ducts. What do you remember about the principal cells? They're the ones that are with the ADH, right, creating the aquaporons. And the The intercalated cells are the ones that are going to help manage those hydrogen ion concentrations and the bicarbonate ion concentrations. And so mainly what we're going to talk about now is how we go through this loop and what happens through the loop when we have lots of water and when we don't have a lot of water. And how we're going to move our water to maintain our blood volume and maintain our blood pressure. As you can see here, we have afferent arteriole coming into the glomerulus, efferent arteriole leaving the glomerulus, and remember there is a capillary bed that's wrapped all around our descending limb of the loop, ascending limb of the loop, and even the collecting ducts. It's all, those capillary beds are all over the place. Notice that the osmotic pressure within the tubule is pretty much the same in the tubule as it is in the blood, but the constituents of that fluid is completely different. Because once we pulled everything out of the tubule and put it back into the blood here, this tubular fluid here, the filtrate, is quite different to the concentration, the osmotic concentration in the blood itself. At the same level, like it's 300 milliosmoles, even though there's different constituents within the tubular fluid and the blood. When that fluid is traveling down this descending limb of the loop, you notice that the osmolarity of that fluid increases, right? It goes from 300 to 350 to 550 to 750 to 900, and then it goes back down again. So how do you explain that? Remember, water is the only thing that can leave the descending limb. Sodium chloride are the only things that are pulled out of this tubular fluid here. So how do we explain this? Anybody got any explanations for me? No? Nobody wants to take a stab? Go ahead, Kesa.
So if the water is leaving on the descending part of the loop, that means that the stuff inside is getting more concentrated.
That's right.
So, yeah. And then when it gets back up, it kind of becomes concentrated the other way because water
can't leave here.
No, yeah, water can't leave, but
Sodium chloride are.
Sodium can.
Yes.
So you add more concentration to the outside
That's
rather
right.
than the inside.
That's right. See this? This is your extratubular concentration of solutes. So as our solutes are traveling down the tube, water is leaving. And why is water leaving? It's leaving because sodium and chloride are leaving here. It's literally the sodium chloride in the interstitial space here is pulling water from here. So that's why we get this concentration. Down, we get to have concentration, then we reduce that concentration and we go back up. Because we are now still pulling from the tubular fluid here. Right? We're still reabsorbing some, not quite as much as we absorbed on this side, but still reabsorbing some on this end, and then we send it down the collecting ducts, and ultimately it's going to become urine. So this is how we maintain our water concentration in our body, through the kidney's ability for counter current multiplication. And this increase in the osmotic pressure, osmolarity of the fluid heading down the loop is the concentration part of that counter current multiplier aspect. This is where the multiplication part comes in. So we are dependent on certain mechanisms to help us manage this fluid variability, right? Sometimes we don't drink as much water as we need to. Sometimes we drink a whole lot more. When we don't drink enough water, what do we need? We need to have ADH levels increased so that we can recuperate, reabsorb as much water as possible. We see that aldosterone is going to play a big role in this, because if aldosterone is involved, then aldosterone is going to increase the reabsorption of sodium and chloride. And when we increase the absorption of sodium and chloride, we increase the reabsorption of water. Wherever sodium and chloride go, water follows. Right. Now that video said the only way that you can move water is through sodium and chloride, but that's not entirely true, because we know that so many different things can be osmotically active, like glucose. Glucose can impact right? How much water is held in the blood because there's so much glucose in the blood, which raises tension, makes you hypertensive, along with that diabetic aspect. So aldosterone causes sodium to be reabsorbed. ADH is going to help get more water into the interstitial tissues and ultimately back into the circulatory system. And so all of these low blood volume, low blood pressure are all going to trigger this hormone complex to work for us. Yeah. So just looking at our kidneys, basically this is happening on a regular basis. So we start out with our 300 milliosmoles out in the cortex, but then those juxtamedullary nephrons are the ones with the big long loop that descends well down into the medulla, right? You got cortex and you got medulla, and you can see that we get this tremendous amount of concentration in those loops as they dip down well into the medulla. And that's what that video was all about, was how the juxtamedullary nephrons are working. He mentioned this specialized set of capillaries called the vasa recta. And the vasa recta is specific for these juxtamedullary nephrons. Whereas we have the peritubular capillaries around the cortical nephrons, these juxtamedullary nephrons with their vasa recta are really all about water moving back and forth. It's a simple, I think of it as a simple mechanism, but of course it's in a complex organ doing lots of different jobs. But you can see here how we go from normal osmolarity to very concentrated osmolarity and then back out to normal osmolarity. And the thing that this is all about is called the countercurrent multiplier and the countercurrent exchange process, and urea playing a big role in being that sponge that draws water into the medullary space and then the water back into the circulation, because we're going to move urea back into the tubule and get rid of it, ultimately. Did you have a question?
I was kind of wondering, this is going to be a stupid question, I feel like. Are the nitrogenous waste also solutes?
All of them are solutes.
Okay.
Everything but water is a
Okay.
solute.
Okay, that's what I thought. I
Yeah.
just wanted to make sure.
Yeah. Solvent, water, solute, everything else.
And so
Solids. Yeah. All right, so how do we establish this huge concentration gradient? Well, we now know, right? We established that concentration gradient through this process of driving the water down through the tubule, through the loop. It's all happening in the loop that we get this concentration gradient going on. And that concentration gradient is what we refer to as the multiplier, because it increases. As we go from 300 down to 1200, back out to 300 and even lower. And that is in the blood flow versus the fluid. So multiplier is increasing in solute concentration. The countercurrent part is because the blood flow is opposite to the tubular flow. Did you notice that? The
Yeah.
blood flow is opposite to the tubular flow. So we have here our salty fluid, but it's not so salty as it is down here, right? Here it's very highly solute concentrated. Water's leaving this descending limb because it can. Nothing else is permeable. It's not permeable to anything else. Sodium, chloride, potassium are pumped out of the tubular fluid here, and that draws the water out from here. It seems a little bass-backwards, but that's the way it's working. And so we have our tubular flow in this direction, but the blood flow is going the opposite way. And this one I don't like. I like these better. Here's our tubular flow. Here's our blood flow. Here's our tubular flow. Here's our blood flow. Opposite direction. So there's the counter-current multiplier system. And the multiplication part comes from starting at 300, ending up at 1200, and back down to 100. How did that happen? Well, we had our tubular fluid get concentrated because water's leaving this side of the loop, and then we have deconcentration, right? What's the opposite of concentration?
dilution.
Dilution, thank you. We have a dilution at this end of the loop because sodium and chloride are leaving and heading toward the blood. And that sodium chloride, while it's in the interstitial spaces, draws water out from this side where water can be drawn. It's not terribly complex, right? You just have to remember that this is the process by which we recover a tremendous amount of water. So those juxtamedullary nephrons dipping well down in the medulla, utilizing that space and all that tissue and holding water there to move it into the blood ultimately, or back into the tubular fluid if we have plenty of water in the system. So this is based on your blood pressure. And your blood pressure is detected by what region of the kidney did we identify as capable of detecting sodium chloride levels by absorbing the tubular fluid? Anybody remember?
I was gonna say the medulla.
It's a little piece of the distal convoluted tubule. It's tucked up into the vascular pole. Begins with an M. There's two names, two words. Those mesangial cells are important for managing the capillary beds.
Yes.
The macula densa. The macula densa, that collection of specialized little cells that are sitting in that distal convoluted tubule right there, and they absorb sodium chloride. If sodium chloride levels are low, then blood pressure is low. Sodium chloride levels are high, then blood pressure is high. Then renin is secreted, or not, right? That renin is going to help raise blood pressure. All right. Question?
Raise or low blood pressure?
Renin is important for raising blood pressure.
Oh, okay.
Because renin is released from the kidney in response to low blood volume. Low blood volume means low blood pressure. That renin then is delivered to the liver, where angiotensinogen is converted to angiotensin 1. Angiotensin 1 is converted to angiotensin 2 in the lungs via ACE, right? Angiotensin converting enzyme. That angiotensin 2 then has widespread impact on all the vasculature, causes vasoconstriction, increasing blood pressure. It impacts the adrenals to release aldosterone. Aldosterone causes the ascending limb of the loop to reabsorb more sodium and chloride. More sodium and chloride reabsorbed means more water, raises blood pressure. Tells the your hypothalamus,
Thirst.
you're thirsty,
Need water.
need water, ADH. So renin is all about low blood volume. High blood pressure, what happens in high blood pressure? Where do we get that signal from? The heart. Atrial natriuretic peptide.
Oh, right.
Where the atria are stretched
Right.
excessively, that hormone then, right, causes to not produce as much ADH, and then we don't retain as much water, and we get rid of that extra water instead of bringing it back into the circulation. Okay, so I'm starting to get a grasp on this. Get a good feeling about the countercurrent multiplier system. Has everything to do with the loop. Has everything to do with making sure that this tubular fluid is the right concentration, and that water is either reabsorbed or not, and that sodium chloride are reabsorbed or not. Because if we don't want to increase our blood pressure, we have to decrease our blood pressure, then we don't want to take in sodium and chloride. We want to get rid of it, pee it out. Along with sodium chloride, what goes? Water. Blood pressure dropped. Because your blood pressure is really, really impacted by your water concentration.
Do we need to know the numbers? Just
Not necessarily. You just need to know how what they represent in terms of concentrations, solute concentration. Yeah. Formation of a concentrated urine. Why would we need concentrated urine? Well, our water volume is low. When our water volume is low, we want to keep as much water in the system as possible, in which case we're going to be forming a very concentrated urine. When you have concentrated urine, how would you know it's concentrated? Just by going to the bathroom.
Yellow.
Super strong yellow color. Yeah, because the darker the color, the more concentrated the urine. When we have water leaving here, we're going to concentrate our solutes. When we have water leaving here, it's leaving because sodium and chloride are leaving this side. And we also want to plug a lot of those aquaporons into our collecting ducts and distal convoluted tubule so we reabsorb as much water as possible. And we produce a very small volume of very concentrated urine. Because that's the way it is. It's the only way to be. Here's this 50% of urine that was filtered, circulates the renal tubes, periodic fluid. Remember that urea really serves as a sponge, like that guy said. It's a medullary sponge. It draws water into the medulla, into those tissue spaces. And then that water can be sent back into the blood or back into the tubules, depending on how much urea there is. And what's urea? Just one of the solutes. What if we have what if we have plenty of water in the system? Here's our concentration of milliosmoles of solutes, and we can see that we don't need to reabsorb as much water and sodium chloride, and so we're pretty able to produce a much more dilute urine because we have plenty of water in the system already. We can really make some pretty tight blood, right? Pretty tight urine to save our blood, that's what I meant to say. Here's some things that impact our ability to form urine. Some valuable, some not so. You can see here, alcohol serves as a diuretic. What's a diuretic? Makes you go. How does your alcohol affect your kidneys? Well, if you don't have ADH released from the posterior pituitary, you're not going to reabsorb all that water that you could if you had the ADH plug those little aquaporons in. This is why, and I don't know if any of you still frequent bars or going out at night like we used to. I never really liked that much, but I mean, you have a long line waiting for the women's bathroom if you're at a bar because you're not absorbing the water. You're just peeing it out because you've got no ADH.
Is that why you get like dehydrated?
You get dehydrated for sure. Yeah, it's a dehydrant. Alcohol is a dehydrant because of that. Yep. And here's the other thing about alcohol. It wets your mouth. Remember one of the first things that tells you, you know, your hypothalamus says you're thirsty. You're too dry. You need more water. So what do? You go and take a sip of something. When you take a sip of something, it wets the mouth. And that's like an immediate tamp down. Okay, hypothalamus, you can chill, take it easy, got a nice wet mouth. But alcohol wets your mouth. That's a little bit of a, right? Sticky wicket there. You're messing with your hypothalamus.
You're messing with
Doesn't
a lot
like
of
to be
things.
messed with. Don't mess with your hypothalamus. So make sure you drink water. Drink water. What else? Caffeine. Caffeine is also a diuretic. Probably you know this because you had a cup of coffee this morning and then on your way to school, you had to pee already, right? Half hour later, you don't drink caffeine.
I don't drink.
So it is a diuretic. And you can see that it inhibits, not only does it inhibit the release of ADH, but it inhibits the reabsorption of sodium. Why do we care about the reabsorption of sodium? Just one second.
Water follows.
Water follows it, right? So when we have sodium being reabsorbed from the ascending limb, then we have water reabsorbed from the descending limb because it's following that sodium. So if you don't absorb the sodium, you got no need to absorb the water. And so you pee more. Yeah.
So like when you're drinking alcohol or even caffeine, they say to drink more water. Is that really even doing anything if you're peeing it all out?
Well, you can dilute your alcohol.
Right.
So you gotta keep up with the water, just so you don't pee it all out.
Okay.
Yeah.
So as long as you're drinking that extra water, it's a little bit better.
It's a
than
little
it
bit
could
better.
be.
Yeah.
Okay.
And you know what? A little alcohol here and there. I mean, honestly, what's life all about? Right? Now, maybe you are an alcoholic and alcohol is not good for you. Maybe you have alcohol that really just messes with your head, then maybe you don't want to do alcohol. But, you know, if you're okay with alcohol, a little alcohol here and there, supplemented with plenty of water. Key is, if you're drinking alcohol, is to have a good solid meal in your stomach before you start drinking, with plenty of lipids. Remember that lipids slow down the absorption of alcohol in the stomach. So caffeine, alcohol, messes with our water levels. Let's look at this. Dilates the afferent arteriole and increases the glomerular filtration rate. What? Why? Can you explain that to me? Oh, we got to review the glomerular filtration rate and the net filtration pressure.
Something to do with like
Push.
What were you saying, Ryan? Yeah, keep it up.
like
If you increase the diameter of the afferent arteriole,
yeah,
then what happens in the glomerulus?
increases pressure.
Pressure. And if you increase the pressure, you're pushing more stuff out, and that is going to increase your glomerular filtration rate. And your glomerular filtration rate, remember, is all about, can you absorb that water? Can you reabsorb the water, or is it flowing through too quickly? So if you increase your glomerular filtration rate, you're going to increase the flow, the speed with which that fluid moves through the tubules, and maybe you can't recoup as much of what you need. You can't reabsorb all those electrolytes that you need to reabsorb.
Which would help with
They get
inhibiting,
flushed right through, right?
which is probably part of the inhibiting reabsorption.
Yes,
Sodium is
yes,
going
yes.
too fast.
Yep, everything's going too fast. So another thing that can happen in our production of urine is this condition called diabetes. And this is not diabetes insipidus, because you know diabetes insipidus has nothing to do with ADH or water or anything like that. Diabetes insipidus is what?
The genes, genetic. No.
And did I say it doesn't have anything to do with ADH? What is diabetes insipidus?
Oh, doesn't it have nothing to do with the insulin?
It has nothing to do with insulin, that's right.
Yeah.
It does have everything to do with ADH. Yeah. Yeah, diabetes mellitus is the one we're talking about right now. I had to look up what diabetes meant. Look, it actually means siphon, and mellitus means sweet, so that makes total sense. And both type 1 and type 2 diabetes mellitus can lead to this sort of concurrent condition called polydipsia, polyuria. Polydipsia, you drink more water. Why? Because you're thirsty. Why are you thirsty? Because you're peeing out all that water instead of reabsorbing it. So polydipsia, polyuria is common signs for diabetes mellitus. Type 1 is the kind of diabetes where you might be born with it or it develops early in life. It has to do with insulin production, insulin receptors. People with diabetes type 1, insulin dependent, usually. Type 2 diabetes mellitus is more of a lifestyle condition, sedentary lifestyle, not enough exercise, not enough use of that glucose, but a diet that provides plenty of readily accessed glucose from high carbohydrate meals, high carbohydrate diet. In which case then you have plenty of glucose flowing and you can't absorb it all, and so it ends up out in the urine. So people with type 2 diabetes can manage their condition with dietary changes, exercise routines, and there are drugs that help with that as well, if you can't get a handle on it. Gestational diabetes is a really interesting one. Anybody here had a baby, developed gestational diabetes? Yeah? You know, it happened to you, Lily?
Yes.
Yeah. And it happens because you have something else growing in you that has a demand for glucose and a demand for insulin, but the pancreas isn't ready to go. And so your pancreas is responsible for producing enough insulin to take care of fetal needs and your own body needs. And the hormones that are causing all this pregnancy, the hormones for the placenta, all tend to add to this process. So you build up a lot of glucose in the blood, and then you don't have enough insulin to handle all that glucose, especially because that insulin is also taking care of the fetus. The pancreas can't keep up. Diabetes insipidus, there's the other one, right? It has everything to do with not enough ADH. This comes to our urine characteristics, which we've already talked about because we did all this in lab. So remember that our pH is typically on the acidic side. This high, that pH of 8 is rather high. During our lab exam, there was that one urinalysis that had a pH of 8. Some people identified that as abnormal, which is true. It's a little on the high side, usually average in the acidic range. Our specific gravity, remember, 1003 to 1030. And this is the measure of solutes in a liquid. We had this, which is not something that we generally refer to, but where does this 1200 milliosmoles come from?
The bottom of the bottom.
The bottom of the loop, yeah, very good. Water content, yep, typically all water, a lot of urine production. This would be, of course, very concentrated urine at 700 mil, and 2000 mil would be a nice dilute, pale yellow color. Clear yellow, pale, wants to be pale. My urologist wanted me to produce urine that looked more like water, because I grow kidney stones. I have a propensity. No bacteria in fresh, clean urine. And we need to talk about how these solutes impact our urine versus plasma. And we talked all about all this stuff. Are there any questions about urinalysis, urine contents? Look, do we have urea in our urine? Yes.
We better.
Absolutely, that's the whole point of it, right? To get rid of those nitrogenous wastes, creatinine, ammonia, and uric acid. Want them all to be in the urine. Do we want to have proteins in our urine?
No.
No, why?
They can be used for other things.
They can be definitely used for other things, but why is it that they don't end up in the tubular fluid? They're too big, right? They can't get through those fenestrations. And what is it that makes the slits?
The
Podocytes?
The pedicels
pedicels.
of the podocytes. Right? The pedicels of the podocytes make the little filtration slits, and the proteins can't get through there either. Do we want to see sodium? Yes, a little bit of sodium in the urine, depending, of course, on our concentration of urine. Right? We don't want to see glucose. We don't want to see glucose in the urine. We don't want to see lipids in the urine. We don't want to see any of these things really in the urine. And then we are, I think, well familiar with all of these conditions. Glycosuria or glucosuria, protein. Don't want to see protein in our urine, but you might if you have a really high protein diet. Don't want to see blood in urine. Maybe in some cases you might have blood in urine. Where would your blood show up in urine?
Menstrual cycle.
Menstrual cycle, exactly. Yep. This is one of the things that we definitely don't want to have show up in our urinary system, urinary calculi. I love this image. Can you believe this? This is what they look like. Ah, no wonder they hurt so bad. Little spiky things. What are they a combination of? They're a combination of, right, all of this. Calcium is the primary contributor for making renal calculi. Yeah. I found out that they name them based on different regions where they're found. The ureteropelvic junction. So ureteropelvic. Remember, there is the pelvis, right where the ureter. So that right there is a place where they tend to get lodged. And where's another one? At the ureteral crossing of the iliac vessels. So remember, the ureter comes down, ureter comes down and crosses over, and there's the iliac veins and arteries. So there's another narrowing, I guess, of the ureters. Yes?
Wait, what do you mean that you find blood in the urine during the menstrual cycle?
If you are menstruating, and perhaps you're not wearing a tampon, and you're using a pad, you might have blood that gets flushed with the urine. Yeah. Not abnormal. It's just that you're on your cycle. You're in your cycle. Right? You're menstruating. Oh, there was another place. The ureterovesical junction, where the ureter empties into the bladder, right? Yeah. Yeah. So they're nasty, very painful. All right, so we've covered the kidney. We're all good with the kidney. I want you to go back because it's been a week. You probably weren't driven to look back at the kidney, but I want you to go back over lectures 1 and 2 and review all of the materials so when we get to lab on Wednesday, we're all up to snuff. We're ready to really look at the anatomy of the kidney. And then we have some histology to look at as well. So where we produce the urine in the kidney, then we have to store that urine because we don't want to be dribbling urine all day long unless we're catheterized, in which case you never have to worry about peeing. Which would you opt for, bladder or a catheter?
Bladder.
I'm going with the bladder. Yeah.
Oh, what do you mean? I definitely want like a whole ass medical equipment just attached constantly.
Dragging a bag around with you all the time. Yeah. No. I
Don't
think
you have
so.
to clean them occasionally too? Or like,
Oh, nasty. Yeah. Nasty, nasty. So, you know, paraplegics,
Yeah.
unfortunately, oftentimes will need to be catheterized because they've lost nerve innervation to the bladder and control of rectum. So we have a structure called the urinary bladder. We have to remember we have a gallbladder and we have a urinary bladder. The urinary bladder is made of a smooth muscle that has its own name. It's called the detrusor. And you're going to remember that. So you're not going to tell me that this is smooth muscle when you're asked what is the name of the muscle of the bladder. You're going to tell me it's the detrusor, right? We have 2 ureters. How are you going to remember you have 2 ureters and 1 urethra? It's one of my favorites. Do you know who urethra Franklin is? R-E-S-P-E-C-T.
I know.
R-E-S-P-E-C-T. That's what you think of me. Hey, suck it to me, suck it to me, suck it to me, suck it to me. Yeah. Okay, you can tell why I never made it as a singer. So we have 2 ureters, and we have 1 urethra Franklin. Get it? 1 urethra Franklin. 2 ureters. These 2 ureters empty into the bladder at these little points right here. These are called the ureteral slits. The ureteral slits, and they are slits because they close really efficiently and they don't allow backflow of urine in the bladder up through the ureters. So the ureteral slits prevent that backflow. Now the bladder wall on the inside has rugal folds, much like the stomach does. The bladder is lined with a specialized epithelium called transitional epithelium. You might see it also referred to as urothelium. Either name is correct. This transitional epithelium is covering the entire inner surface of the urinary bladder. The rugal folds, however, do not cover the entire inner surface of the bladder. There is a triangular region called the trigone that is smooth. No folds, no rolls. What do you think is the reason for that? It's a cone shape. So remember, we have a 3-dimensional structure, so it's a cone shape right at the base leading to the urethra. Why is that valuable?
For like
Urine
exit,
out?
proper exit of urine, or like funding.
Making sure that urine flows, that it leaves the bladder, it doesn't get hung up in those folds. Exactly right. So the entire inside lining of the bladder is transitional epithelium. Much of the bladder wall is covered with rugal folds because now we can fill the bladder up with urine and then empty the bladder, and you fill it up and empty it. But we want the trigone between the ureteral orifices and the urethra. We want that to be nice and smooth, making sure that the flow is regular, nice, easy flow. The ureters are lined with transitional epithelium as well, and the urethra is lined with transitional epithelium in females. In males, it goes through some transitions, which we'll talk about in a moment. This is an image of the female's urinary bladder and urethra, and you'll notice that there is no internal urethral sphincter here on females. It's a thickening of the detrusor that closes the urethra at the bladder end. There is an external urethral sphincter just above the external urethral orifice. And that urethral sphincter, as you can see here, is part of this muscle that forms the floor of your pelvis, this urogenital diaphragm. This is the male's version. Here's our urinary bladder, 2 ureters, 1 urethra. Trigone, smooth. Ureteral slits defining the edges of the trigone. In males, we do have an internal urethral sphincter. We have a prostate gland that wraps around the urethra. Maybe you've heard of this. Older men, 50, 60, 70 years old, prostatic hyperplasia is a very common problem. And when the prostate hyperplasias, in other words, it gets bigger, it can impede the flow of urine via the urethra because it occludes, it can close that urethra down, which is why older men are up and peeing 4 or 5 times a night because they can never really empty their bladder. So the bladder is always going to feel pressure because they can never really completely empty it unless they have something done to their prostate to reduce the size of the prostate and sort of remount that urethra. Then we have a membranous urethra right here where our external urethral sphincter is located. So prostatic urethra, membranous urethra, and then the spongy urethra all the way down the length of the penis. And so our penis has a dual purpose in males. One is to deliver urine from the bladder to the exit. The other is to deliver sperm. And we're going to talk about how that happens. In fact, this is probably the opening of what's called the ejaculatory duct from the seminal vesicles into the prostatic urethra through the prostate. We have transitional epithelium here. Then we go from transitional in the prostatic to pseudostratified columnar in the membranous, pseudostratified columnar down to columnar via the end of the penis, epithelium-wise. Did you know that this is the official term for peeing? Micturate. When you micturate, you pee.
You had it in a previous slide.
Hmm?
You had it in a previous slide,
Yeah.
which is the only reason I know.
Micturition reflex is the next thing to talk about. Which of the 2 systems, sympathetic, parasympathetic, is in command here?
Parasympathetic.
The parasympathetic. That's
Sympathetic
right.
seizes you up.
Because you don't want to have to be peeing, so our sympathetic system is not involved here. The parasympathetic is the one that is going to be stimulated to relieve the bladder. Our bladder is great in that it can hold a whole lot of urine. You can feel that pressure that is exerted on the walls of the detrusor muscle, and you have parasympathetic fibers, sensory fibers that are going to go up to the pons, from the pons all the way up even to your cerebral cortex. Because while you feel that pressure in your bladder, are you ready to go pee at that moment? Not necessarily. So we can override this system just like we could override it from our rectum, right? We didn't have to go defecate right off the bat, necessarily, unless it was diarrhea. And then, but in the bladder, you can really fill your bladder right up and hold it for a while. Ultimately, however, the sympathetic system, the parasympathetic system is going to win out. It's going to drive the emptying of that bladder because you can't hold it forever. This is what happens with babies. They have no idea what that pressure means. They just let go when they need to let go. But you're teaching that 2-year-old what that pressure feels like and what you do about it. Off to the toilet. Yeah. So that's recognizing the pressure from the parasympathetic fibers. Remember in the pons we had that urine pontine, you know the time I'm looking, pontine nuclei?
Yeah.
It's the urinary pontine nuclei in the pons that helps us manage our bladder control. These sphincters, external urethral sphincters, are a skeletal muscle. Just like our anal sphincter, external anal sphincter was skeletal muscle, These two are skeletal muscle, which means that we have voluntary control over them. And we learn to relax those muscles because they're always constricted to release pressure from the bladder. Yep.
So when some women have babies and their pelvic floor becomes weak, is it that muscle or is it?
The whole thing right there.
Oh, okay.
Yep. Have you heard of Kegel exercises?
Yes.
Squeezing. Keep practicing. Keep practicing.
So it
Very
never has anything to do with
The internal.
the
Nope.
trigone?
No, it's the external.
Okay.
Yeah. Yeah. All right. Yep, it doesn't matter. If your bladder's full enough, you're gonna pee. That's it, guys. That's all we need to talk about for this.
Do we need to know that wobble?
The
That whole thing?
full bladder. So you need to know about parasympathetic innervation
Okay.
primarily. Yeah. Because that's what you're stimulating. And that's what your body's telling you.